Okay, no problem. So, quick outline. I'll go through some background information, the capabilities we have in our imaging group in Boston, and then sort of the rationale behind and the status of our collaboration with inviCRO. Then I'll spend the bulk of the time talking about an ALS pilot study that we did using the inviCRO platform in terms of the data acquisition, our analysis and storage, and then the state of our collaboration moving forward. So Merck's imaging group, across our network, we have groups in Pennsylvania, Singapore, Boston. In the Boston group in particular, where I'm at, we have a 7T MRI from Bruker, 2 IVIS bioluminescence imaging machines, 2 micro CTs, GE Locus Ultra, and Explore CT120, as well as a Vivo 2100 ultrasound. Today I'll be just touching on the MRI for the purpose of this presentation. So the rationale behind collaborating with inviCRO. So this is something that Scott Ziegler in our West Point, Pennsylvania group has really spearheaded, but he identified that we have quite a few machines across the network, 6 machines in Boston that generate a massive quantity of data, and that having them stored on individual servers puts data at risk, and it's not very cost effective or efficient. And so the solution that he came up with was to partner with inviCRO and use a cloud-based storage system here at the iPACS for our analysis, and with the goal being have the iPACS sync to a GMAS NRD storage system for long-term storage in order to have a more cost-effective and efficient system. And the idea here is we can query the data more easily. It improves our archiving from a compliance perspective. We can just input links into our lab notebooks. Our data retention is better. And so it started out as a 5-year plan. Now we're into the 3-year plan, which is to have, like I said, the data automatically sync from our machines to the iPACS to a long-term storage. All right, so for the rest of the time I'll talk about an ALS mouse model that we've been working with in Boston, and how we used the iPACS system to validate an MRI biomarker in this model. So ALS, if you're not familiar with it, is a rapidly progressing neurodegenerative disorder that's characterized by motor neuron death, both upper and lower motor neurons. And one of the first mutations that was identified in humans about 20 years ago was the superoxide dismutase 1 protein, commonly a G93A mutation. This mouse model's been around for about 20 years. It recapitulates several components of human ALS. The mice have progressive motor impairment, usually measured by rotarod. However, there are some caveats. There have been compounds that worked in the mouse model that didn't work in humans, so we want to be really careful and try and get as much information as we could out of any preclinical studies. Another caveat with these mouse models is you have really large N per group, and they're very long studies. So, longitudinal motor behavioral assessment by rotarod, it's highly variable, and so you need to have large numbers per group. And oftentimes people use a peel-off design and use terminal immunohistochemistry as a way to track motor neuron death. But we found a promising publication that showed that you can use MRI to track changes in the brainstem of these mice, and this is getting at more of the bulbar symptoms of ALS. So some patients present with a more rapidly degenerating, speech-impacted version of the disease. And so we wanted to validate this biomarker looking at brainstem degeneration using exclusively the iPACS VivoQuant platform. So I'll walk through this slide briefly, but the kinetics of the mouse model, in the top left in panel A, you can see a rotarod measure. This is mice are placed onto a rotating rod, and it accelerates, and over time they fall off the rod. So the time it takes them to fall off the rod is plotted basically over time. And the top line there is just wild-type mice. You can see the mutant mice show a progressive decline in their ability to perform this task, significant by day 100 and rapidly declining by day 120. This is accompanied by a drop in body weight, which is also significant at day 100. But this paper that we wanted to replicate showed that in these mice that they considered pre-symptomatic, you could pick up changes in the brainstem motor nuclei, specifically the trigeminal, facial, and hypoglossal. And these three nuclei are involved in things like facial movement, tongue movement, swallowing, and in human speech. Mice can't speak in the same way we can, of course, but so they went in and they performed just a manual analysis of just the voxel intensity in these three nuclei, and they plotted at the very bottom for each nucleus a significant increase from wild type in hyperintensity. So they're taking a manually drawn region of interest around the brainstem and comparing it to a control region from the same slice. Now, before we started doing any scanning, we wanted to understand what exactly does it mean to have a hyperintense region of your brainstem, and luckily they looked into that. So with a T2 weighted MRI scan, the water content, if there's increased water content, you'll have higher intensity. And so they show in this paper, and others have shown in this model, that there's increased vacuolation, likely a result of mitochondrial defects and cell death in these motor neurons, as well as increases in GFAP expression and IBA1 expression, which are indicative of neuroinflammation. And all three of those metrics were highly correlated with this T2 signal. And here is just showing what we're trying to replicate, these three nuclei, as you can see visually compared to the wild-type mice on top. They have hyperintense regions. So as far as our study design, we went forward with 10 wild-type mice and 10 G93A mutants. We did some standard metrics, took the body weights at a weekly basis, as well as rotarod and locomotor activity. And we also performed this T2 MRI scan from the paper at days 40, 60, 80, 100, and 115. We used, as I said, VivoQuant and iPACS exclusively for the analysis and data management. And the first consideration that we needed to look into was having a more consistent naming convention. And just briefly, so we were somewhat constrained by the data fields that we had available from the Bruker Paravision system. And so on the left are the instrument fields, on the right are the naming conventions we decided on. In particular, I'm highlighting the mouse ID because that proved to be somewhat of a challenge early on. So we used the iPACS sync tool to transfer the data from our local store up into the iPACS. Initially, we had some difficulty that there were some redundant file names coming off the Bruker computer, and that required us manually transferring them and then renaming them. Suffice it to say, TJ was very patient working with me and doing some editing of the indexing configuration files, which was helpful and allowed us to sort by mouse ID in the web browser. And as I said, this was my first foray into data management, so I can't thank the crew at inviCRO enough for being patient with me. Just a screenshot of what it looks like accessing the same data on the iPACS web browser as well as in VivoQuant. And we'll go into that a little more in a moment. So as far as the analysis method we used, again, the goal was to non-invasively quantify the degeneration of these brainstem motor nuclei. And so similar to the paper, we used an online available mouse brain atlas and identified the trigeminal, facial, and hypoglossal nuclei. And then using just example scans at the bottom, these are T2 weighted scans, and highlighting in red the trigeminal, in dark blue the facial, and in purple the hypoglossal nuclei, and then comparing to a control region within the same slice to get this T2 hyperintensity value. All right, I'll just quickly walk through my workflow. It's a lot more simple than what TJ was just showing us earlier. But basically, we open up the VivoQuant software, click on, do I have a cursor here? No. Click on the data browser, identify the scan that we want and the time point we want, click open, data pops up, manually adjust the visualization threshold, and already the trained eye can start to see the trigeminal. Go into the 3D ROI tool, generate those ROIs that we want, and here there are going to be 6 of them, the 3 nuclei and their control. And then just manually place the regions of interest and the control regions. There's the facial and the hypoglossal. And then we can go in, save that right to the iPACS, and then from there we can log into the iPACS to generate a data report, export the data, and come up with all the values we want for the entire study. Here, just showing the same mouse, either wild type on top or G93 on the bottom. This is at 15 weeks, so a terminal time point. You can see the wild type mouse has just about no hyperintense regions in the brainstem, versus the mutant mouse has visually apparent hyperintense regions for all 3 nuclei. And then quantitatively, going through, you can see for all 3 nuclei there's a significant and stark difference between the wild type and the mutant mice. So you know, to us, this was a successful replication of what we had seen in the literature, and it was we felt good about using the iPACS and VivoQuant exclusively in this case, and we were ready to move forward with, you know, additional studies now that we felt that this biomarker had been validated. Quickly, as far as compliance, you know, we can just take the analysis we did and the data that we have, we can log into the iPACS, copy the address into our notebooks, and that's good for archiving. And as far as future directions, as I mentioned, the goal is to have the data flow from the machines to the iPACS for analysis, and then sync automatically to the long-term storage. We're manually migrating the old data, and we're looking forward to using VivoQuant in place of Amira. So with that, I want to make sure and acknowledge everybody on this slide, in particular the folks on the left. TJ, as I said, extremely patient on the inviCRO side. Chris and Diane in the neuroscience group were instrumental in getting this project started and working with the animals. Richard Kennan in our group in Kenilworth, who helped design the MRI protocol. Raquel Sevilla, who helped show me how to use the VivoQuant. Amy Vanko, who's our point person for the iPACS, and Scott Ziegler, who, you know, this whole collaboration with inviCRO was his brainchild. So with that, I'll take any easy questions you might have. Anything online?
Yeah, we have a question online. If you could go into a little bit more about after the VivoQuant, where you know, from the VivoQuant to those plots, what happened in between?
Okay, so I saved all the ROIs, the nuclei, and then its respective control to the iPACS, and then I just logged in, generated a report, selected that I wanted the mean intensity of the region of interest, and then exported that to Excel, created the equation, created this equation here at the bottom, and then plotted it. So pretty straightforward.
So is your ROI drawing based on the 2D image?
Yes.
We also had another call online. You mentioned briefly the metadata with the importing. I think when you were talking about the iPACS sync,
Mm-hmm.
looks like they were just asking if you could go into that a little bit more. What was necessary there?
That might be a good question for you to answer, because that's I got some of this slide from you, but yeah. So I think just it was helpful. What we wanted was to be able to basically sort the data by the time point and the mouse ID, and I think you had done some work behind the scenes, being able to grab that that field from the Paravision software where we input the mouse ID and having that visualized on the data browser. Yeah, beyond that, I'm not sure what you did. You did some computer magic behind the scenes.
Thanks, Dan.
Yeah.
So by computer magic, he means that any of the metadata fields can be remapped to any database object that we're indexing, so that you can parse over what logically makes sense to you, like the NYD.
Exactly.
So,
Thanks.
all right, thanks, Kane.